Today we are going to discuss on the megasporogenesis, its structure and types of ovule. In last lecture we also discussed flower. Flower is nothing but the modified shoot which is developed from the accessory whorls to modified into reproductive whorls. It is nothing but the floral primordia from which the flower has been developed, and this primordia develops first into the floral buds and then into the flower. It is actually the plant's reproductive part, where we can find the androecium represents the male gamete, whereas the gynoecium represents the female gamete. So here you can observe that typical flower has been shown here. Here you can observe the typical gynoecium. Here you can find this ovary, and inside the ovary there is the development of ovule. Then what is actually the ovule? How it develops? The ovule develops from the multicellular placental outgrowth, including the epidermal and the number of hypodermal cells. This is the typical structure of the angiosperm ovule, where you can find at the center there is the development of megaspore has been taken place. This is the micropylar opening, and this is the funiculus. This is the typical structure of the ovule of angiosperms. So it is developed from the multicellular placental outgrowth, including the epidermal and the number of hypodermal cells. This gives rise to nucellus and one or more integuments from the basal region. So these are the integuments which are developed here, and these integuments are developed from the epidermal and the hypodermal cells further. In ovule, we find there are two integuments. Usually the inner one forms the first and the outer one is the first, then the outer one. So here inner one is more delicate. The जास्त नाजूक आहे inner one, whereas the inconspicuously developed than the outer one. Here the outer one is quite sturdier and helps in the protecting the megaspore inside, and the inner one is the delicate one. Here you can find one hypodermal cell, not epidermal, but the one hypodermal cell which is below the epidermis. Now here you can find, here it is the typical epidermal cells. The inner cell is the hypodermal cell, which becomes quite bigger in size, and here you can find the cytoplasm becomes the dense, and the conspicuous nucleus can be observed here. So this is now called as a megaspore mother cell, or we can say the archisporial cell. This archisporial cell divides transversely. Here you can find the divisions has been taken place, divides transversely and forms the inner primary sporogenous cell and outer primary parietal cell. Now here the outermost layer is developed. This cell is nothing but the parietal cells, and the centrally you can find there is the development of the sporogenous cells inside. So these are the dyad stage where you can find there is the transverse division of the cell is taken place. And there is the development of the first sporogenous cells where you can find the transverse division is taken place, and the another divisions which are the vertical divisions where you can find there is the development of the parietal cells is being developed. The lowermost cell is nothing but the megaspore cells. Now these megaspore mother cells sometimes does not get into the divisional state, but they can directly act as a megaspore mother cell. When the megaspore mother cell divides, it becomes, now it is a diploid cell. After fertilization, there is the 2n number of chromosome present there, which undergoes meiosis and forms 4 haploid megaspores. This diploid cell gives a haploid 4 megaspores. Okay? Out of these 4s, only one, here you can find that, only one remains at the chalazal end and behaves as a functional megaspore. Now only one can be act as a functional megaspore, and others which are present at the chalazal end toward the micropylar end, they gradually degenerates. म्हणजे जे 4 इथं स्पोर तयार झालेले आहेत हॅप्लॉइड, आउट ऑफ विच 3 gets degenerated and only one functional megaspore is remained there at the chalazal end. And the functional megaspore forms the female gametophyte, or we can say the embryo sac. Then megaspore, which is haploid in number and is the first cell of the female gametophyte. आता हा जो इथं मेगास्पोर तयार झालेला आहे, इट इज द फंक्शनल मेगास्पोर. आता हा जो हॅप्लॉइड स्टेट असलेला स्पोर आहे, इट इज द फर्स्ट सेल ऑफ द फिमेल गॅमेटोफाइट. अँड इनिशियली दिस एम्ब्रियो सॅक इज युनिन्युक्लिएटेड, जी आपण लास्ट डायग्राम बघितली, अँड फर्दर ग्रोथ ऑफ इट न्यूक्लियस डिवाइड्स बाय 3 successive divisions to become the 8 nucleated stage. Here the first division, this one is the second division, and this is the third division, where you can find the 8 nucleated stage has been developed here. Now this end is the micropylar end, where you can find the 4 nuclei. They stay there at the micropylar end, and 4 shifts towards the chalazal end. So this end is the chalazal end, where you can find the 4 nuclei remains there. म्हणजे वर 4 आहेत, खाली 4 आहेत. त्यापैकी खालून एक मायक्रोपायलर एंड कडून एक येणार आणि दुसरा चलाजल. आता मायक्रोपायलर एंड कडून एक न्यूक्लियस सेंटरला गेला आणि चलाजल एंड कडून एक न्यूक्लियस गेला आणि मायक्रोपायलर कडून एक आला. अशा पद्धतीने सेंटरला 2 न्यूक्लियाय तिथे एकत्र आलेत. सो इट फॉर्म्स अ पेअर ऑफ polar nuclei. Now these 2 nuclei are called the polar nuclei. These nuclei fuse together to form a diploid nucleus. ठीक आहे, आता यांचं इथं फ्युजन होणार आहे, and thus it forms the diploid nucleus. It is the definitive nucleus and it is also known as secondary nucleus. ठीक आहे, सेंटरला जो दोन्ही बाजूने चलाजल एंड कडून आणि मायक्रोपायलर एंड कडून जे न्यूक्लियाय आलेले आहेत, त्या दोनचं फ्युजन होऊन इथे एक डिप्लॉइड न्यूक्लियस तयार झालेला आहे, अँड दॅट डिप्लॉइड न्यूक्लियर इज नोन एज द सेकंडरी न्यूक्लियस. द 3 nuclei which are at the micropylar end and egg apparatus, here you can find that these are the micropylar end. Here at the micropylar end 2 synergids and the egg cell, these 3 nuclei are there, and here at the center you can find the polar nuclei. So these 2 cells जे आता chalazal end ला आहेत, those who are at the chalazal end, they are now called as the antipodal cells. So these antipodals at the chalazal end and synergids are at the micropylar end. The egg apparatus in each nucleus is surrounded by the viscous mass of the cytoplasm. It is normally without wall, and thus in the middle one is the largest, and this cell is called the egg cell. Here you can find these 2 synergids and the third one is the egg cell. We also call them as the ovum, oospore or egg. The rest 2, which are known as the synergids, and these are nothing but the supporting cells to the egg cell. The antipodal cells have the viscous mass and the cytoplasm covered by the cellulosic wall. Here you can find that these 3 antipodals are present at the chalazal end. Now this is about the development of the particular embryo of the angiosperm. म्हणजे एका सेल पासून, एका न्यूक्लियस पासून त्याचं डिव्हिजन झालं. 3 stages of division has been taken place, and from 2, 4 and 8 cell structure has been developed, and after 8 cell structure 4 moves to the chalazal end and 4 moves to the micropylar end, where we find out of these 4, 1 from each end comes in the center and it forms the polar nuclei, which is diploid in nature, and thus there is the formation of the embryo is taken place, where egg remains at the micropylar end and it is supported by the synergids. आता आपल्याला अभ्यासाला जो सॅक आहे, it is the monosporic type of the embryo sac development. ही monosporic development कशी होते याचा आपल्याला अभ्यास आपल्याला इथं फर्स्ट इयरला करायचा आहे. This type is also known as the Enothera type of the embryo. This embryo sac is the monosporic type and also known as the Enothera type, because in Enothera this type of the development has been observed. It is also known as the polygonum type of the embryo sac. It is usually linear tetrad of the megaspore and which are formed, but instead of the innermost one, the outermost megaspore, which is present towards the micropylar end, remains functional and forms the embryo sac. आता याच्यामध्ये आपल्याला ही गोष्ट लक्षात ठेवायची आहे की जो आउटरमोस्ट मेगास्पोर आहे, जो मायक्रोपायलर एंडला आहे, दॅट रिमेन्स फंक्शनल अँड अदर्स गेट डिजनरेटेड. आता द फंक्शनल मेगास्पोर अंडरगोज 2 सक्सेसिव्ह डिव्हिजन्स. आता ही जी डेव्हलपमेंट आहे ती आपण जस्ट आता डिस्कस केलेली आहे. द फंक्शनल मेगास्पोर अंडरगोज 2 सक्सेसिव्ह डिव्हिजन्स अँड द forms the 4 nuclei. All the nuclei remains towards the micropylar end. Out of these 4 nuclei, 3 nuclei forms an egg apparatus and egg and 2 synergids, and the remaining one forms the single polar nucleus. Out of 4 nuclei, काय होते याच्यामध्ये 3 nuclei forms egg and egg apparatus, ज्याला आपण egg आणि 2 synergids म्हटलेलं आहे, आणि जे रिमेनिंग वन आहे तो काय करतो, इट मूव्स टू दी पोलर न्यूक्लियस और मूव्स टू दी सेंटर. देन सेकंड पोलर न्यूक्लियस आर द अँटीपोडल सेल्स विच आर ऍबसेंट. इनोथेरा मध्ये काय आहेत, सेकंड पोलर न्यूक्लियस अँड द अँटीपोडल सेल्स आर द ऍबसेंट इन द इनोथेरा अँड अदर मेंबर्स ऑफ द ओनॅग्रेसी फॅमिली. So here you can find in the diagram this type of the structures is being developed. Here you can find a single cell, a single mass of cell where one cell get recognized as a archisporial cells. This archisporial cells now transformed into an megaspore mother cell, and this megaspore mother cell further get divided. आता इथे आपण 4 सेल्ड स्ट्रक्चर तयार झालेला आहे. दिस 4 सेल स्ट्रक्चर फंक्शनल मेगास्पोर जो आहे तो इथे आपण दाखवलेला आहे. दिस फंक्शनल मेगास्पोर मध्ये 2 न्यूक्लिएटेड स्टेज हॅज बीन डेव्हलप्ड अँड दिस 2 न्यूक्लिएटेड स्टेज 4 न्यूक्लिएटेड स्टेज. आता एकच फंक्शनल आहे. ह्या 3 जे न्यूक्लियस आहेत दे गेट ऑटोमॅटिकली डिजेनरेटेड. After degeneration, only one functional megaspore remains, and this megaspore get divided into 2 at first stage. Then at second stage, we find there is the second division where 2 is get into 4 nucleated structure. So it is the 4 nucleated structure, and these 4 nucleated further divide to get the 8 nucleated structure. Now this 8 nucleated structure where you can find 4 are shifted to the chalazal end and 4 are shifted to the micropylar end. आता ह्या दोन्ही पैकी जे 4 डिवाइड झालेले आहेत, वन ऑफ दिस 4 मूव्स टू दी सेंटर फ्रॉम ईच साइड. म्हणजे चलाझल एंड कडून सुद्धा एक न्यूक्लियस सेंटरला आलेला आहे आणि त्याच वेळेस मायक्रोपायलर एंड कडून सुद्धा एक न्यूक्लियस आपल्याला तिथं सेंटरला आलेला आहे, विच इज फ्यूज टू फॉर्म द डिप्लॉइड न्यूक्लियस और सेकंडरी न्यूक्लियस. याला आपण काय म्हणतो, इट इज द पोलर न्यूक्लियस. ठीक आहे. आणि आता याच्यामध्ये आपण बघतो की अँटीपोडल जे आहेत दीज आर ऍट द चलाझल एंड अँड द मायक्रोपायलर एंड हॅव बीन शोन द एग अँड द सिनर्जिड्स. तर अशा पद्धतीची डेव्हलपमेंट ही आपल्याला मोनोस्पोरिक टाईप जो एम्ब्रियो सॅक आहे, ज्याला आपण पॉलिगोनम टाईप ऑफ द एम्ब्रियो सॅक म्हणतो, तर अशामध्ये ही डेव्हलपमेंट आपल्याला झालेली दिसते. सो दिस इज द डेव्हलपमेंट ऑफ द मेगास्पोर फ्रॉम द मास ऑफ सेल्स अँड अप टू द डेव्हलपमेंट ऑफ द angiospermic embryo, particularly of monosporic polygonum type of the embryo sac develop. So there are various types of the ovule development. So we are going to start with the types of ovules now. The first type in the ovule development is the orthotropous type of the ovule development. Ortho means a straight and tropous means turn. So there is no turning at all. So it is the straight development of the ovule and it is known as the orthotropous or the atropous development of the ovule. When micropylar and chalazal and funicle lies in one straight line. So this is called the orthotropous or the atropous development of the ovule. So it is where we can find micropylar end, chalazal end, and the funiculus. They are all in one line, and because of that it is known as the atropous ovule or the orthotropous ovule. It is found in the family Polygonaceae and the family Articaceae. The second type is the Anatropus Oval. Ana means the backward and tropus means turn. So here you can find it is a completely turn as compared to the Orthotropus Oval. A complete turn of this ovule is taken place, and thus it is called the Anatropus Oval. So it is completely turned back into 180 degree. So here you can find the body of ovule turns backward completely with the angle of 180 degree, and so the micropylar end becomes close to the hilum. So here you can find it is more closer to the hilum and the placenta. Then Hemitropus oval. Hemi means half, hemisphere आपण म्हणतो. So it is hemi means the half, and the tropus means turn again. So here you can find there is a turning of ovule is taken place, and the body of ovule is placed transversely, almost transversely, or somewhat right angle to the funicle. So this is the funiculus area where you can find this is the completely right angle to the funiculus side, and the chalazal and micropylar ends are present in one straight line. Here you can find the micropylar and chalazal end. Here they are at one, but the funiculus, it is right angle to the funiculus. So it is a straight line where we can find, and it is found in the ranunculus, where this type of the ovule is also known as the ranunculaceous type of the ovule. Then another type is the Campylotropus. Campylos means curved. So here you can find it is the slightly curved type of the ovule is taken place here. Here the body of the ovule is curved in such a way that the chalazal end and the micropylar end not lie in the same straight line. आता याच्या आधीच्या आपण याच्यामध्ये बघितलेलं होतं. Here you find micropylar and chalazal end are in one line. Even in Anatropus oval, they are in one line. Even Orthotropus, they lie in one line, where chalazal end and the micropylar end, they are at one line. But here in Campylotropus, we find there is no straight line can be found, and thus chalazal and the micropylar do not lie in the same straight line. This type of the ovule is normally found in the family Leguminosae and known as the Campylotropus ovule. Then Amphitropus. Amphi means both ways. Now here you can observe that here is the curvature of ovule is more pronounced, and the embryo sac becomes the horseshoe shaped. म्हणजे घोड्याची जी नाल असते त्या पद्धतीचा हा एम्ब्रियो इथे आपल्याला डेव्हलप होताना दिसतो. Because of this horseshoe shaped structure, it is become the Amphitropus. So here also you can find there is no chance to get into one line, but we can find horseshoe shaped type of the embryo development is found in this type of the ovule. So it is the Amphitropus ovule found in the family Butomaceae. The Cersinotropus. Now here you can find the funicle is very long. This completely structure, funiculus end is long, and you can find the ovule rotates almost in 360 degree angle, and in such a fashion that it is completely circled around the funicle. And because of that, this micropyle faces upward, the micropylar end faces upward, and this type of the particular ovule is found in the family Cactaceae. So it is the Cersinotropus, means the almost circular type of the embryo, and here this type of the ovule is known as the Cersinotropus ovule. Here you can find some of the basic structures that we should know It is the LS of Anatropus Oval, and every type of the ovule you can. It is attached to the placenta with a stalk called the funicle. This complete structure, a backbone-like structure, is known as the funicle, and which is the vascular supply inside. So this part is known as the funicle. The point of attachment of the funicle with the body of the ovule is known as hilum, with the extending above in the form of a ridge-like raphe. This portion, this particular gap is known as the hilum. So this is the point of attachment of the funicle with the ovule. So it is the hilum portion which can be extended in the form of a ridge-like structure. Then nucellus consists of a parenchymatous cells. So here you can find the central portion, nucellus, and this nucellus consists of the parenchymatous cells. These are all parenchymatous cells here inside. Then nucellus is remain covered by one or two coverings called integuments. Here you can find this yellow line. In this yellow line you can find these two are the integuments, and it is mostly covered by the integuments. These integuments remain discontinued at the end of the one of the point, forming a passage called the micropylar end. So where there is the opening, this opening is known as the micropylar end, and this end is known as the chalazal end. So base is nothing but the chalazal end, and opening is there, that end is known as the micropylar end. So embryo sac consists of 3 antipodals, 2 synergids, and 1 egg cell. Here you can find these 3 are nothing but the structure of the ovule. At the center we find diploid polar bodies. Here at the chalazal end we find the 3 structures. We can say that these are the antipodals, and here you can find 1 is the egg cell and 2 are the synergids at the micropylar end. The antipodals are located near the chalazal end, and the egg cell and synergids are towards the micropylar end. So here at the micropylar end we find the egg cell and the synergids. Here 1 is the embryo sac, where you can find this is the 1 is the egg cell and another 2 are the synergids, whereas at the chalazal end we can find there is the presence of the antipodal cells.